Hello students, welcome to EPG Pathshala. I'm Dr. Saima Amin from Jamia Hamdard. Today we are going to talk on module Evaluation of Liquid Dosage Forms under the paper Pharmaceutical and Biological Analysis. After understanding the module related to this dosage form, the learning objectives will be the students will be able to understand the problems which are encountered in a liquid dosage form and how these problems are evaluated for a liquid dosage form. Coming to the introduction of this module related to liquid dosage forms, first of all, we need to understand what are the liquid dosage forms. These are the oral pharmaceuticals which are available in a liquid dosage form, and these liquid dosage forms include all the monophasic formulations such as solutions, syrups, aromatic water, elixirs, spirits, concentrates, and all these kind of monophasic formulations offer better patient compliance as they are more palatable and execute prompt effect. In a liquid dosage form, the molecules of the solute are dissolved uniformly into the solvent, and many of these are simply the solutions of medicament in water, alcohol, ether, glycerine, oil, or chloroform. Liquid dosage forms are generally formulated for geriatric or the pediatric patients. These dosage forms offer variety of advantages such as no dissolution time is required for such dosage form, and thereby rapid absorption is anticipated from the intestine or the stomach as compared to other kind of dosage forms. Therefore, these formulations are designed for the ailments where we require prompt efficacy, for example in case of pain and fever, but there are chances that the high plasma concentration level for drugs can be achieved from such a dosage form, which could be harmful also. Liquid formulations require various excipients in order to be available as a composition, and the excipients which are incorporated in these dosage form include the vehicle, solubilizer, stabilizer, and the viscosity modifiers or viscosity imparting agents also. At times, we need to incorporate preservatives also, sweeteners and colorants and flavorants also for the stability of the formulation and to provide, you can say, organoleptic properties to the formulations. The drug's physical and the chemical characteristics such as the solubility, pH stability, pK value of reactive functional groups are essential in order to choose the proper excipients. Since most of the liquid dosage forms are aqueous formulations, therefore, potable water is used as a vehicle for these compositions. As per United States Pharmacopoeia, the potable water configuration indicated in this pharmacopoeia states that the water must have the pH in the range of 5 to 7. The total organic carbon less than 500 parts per billion must be present. Then total bacteria count should not be more than 10 colony forming units per milliliter and must be pathogen free. But the level of endotoxin is not defined, as it is the requirement for this sterile water for injection or water for injection required for parenteral formulations. The conductivity and resistivity of the potable water should also be measured at variable temperature ranging from 0 degree to 40 degree centigrade. These dosage forms are intended for internal, external and parenteral administration, as we all know, and therefore can be supplied in multi-dose or as a single dose preparations. Usually, a metering device is supplied for a multi-dose container for oral liquid dosage forms, and the device is usually a spoon or a cup of volume of around 5 to 10 ml or an oral syringe or a suitable dropper also for ear drops. Some examples of oral dosage forms include ephedrine, elixir, codeine, lintis, and acid gentian mixture, orange flower water, cinnamon water, spiritus frumenti containing around 47 to 53% of alcohol and is prepared from the cereals. Mostly syrup, which denotes a solution containing high proportion of sugar or sucrose, is used as a vehicle for its sweetening and flavoring properties. For example, we have a blackcurrant syrup available, lemon syrup available as flavorant. Intravenously administered solutions include normal saline, ringer, lactate solution, and other categories also. For example, for external administrations, formulations like lidocaine, topical solution, or iodine solution are also applied on the skin. Evaluation of liquid dosage forms. The first evaluation parameter is uniformity of content. The content of active ingredient from the randomly picked 10 containers of a liquid dosage form is taken. The formulation complies with the test if the individual value of each container lies between 85 to 115% of the average value. The formulation fails to comply with the test if more than one individual value is outside the limits of 85 to 115% of the average value, or if any one individual value is outside the limits of 75 to 125% of the average value. If a single value is outside the limits of 85 to 115%, but within the limits of 75 to 125% of the average value, the test is repeated using another 20 containers taken randomly. The formulation complies with the test if in the total sample of 30 containers, not more than 3 individual values are outside the limits of 85 to 115%, and not more than one is outside the limits of 75 to 125% of the average value. The test is same in Indian Pharmacopoeia, British Pharmacopoeia, and United States Pharmacopoeia. Out of the different parameters which are required for quality assessment of such liquid dosage form, the one is the visual inspection, where for visual inspection of these liquid dosage forms, sign of physical instability is observed, which is indicated by either formation of lumps or floccules or sediments or crystals. A change in color may indicate chemical degradation or microbial contamination of such formulations also. With visual examination or inspection, the ingredients and the final products are carefully examined for purity and the appearance. The physical evaluation for appearance is required for patient adherence also and compliance. Therefore, the formulation should be clear and free from any kind of precipitate. If the refractive index which is similar to the if the refractive index is similar to the refractive index of water, which is 1.33, and the percentage transmittance is above 90%, then we usually say that these liquid dosage forms are clear or transparent. The result of percentage transmittance, refractive index, and turbidity suggest the clarity of the formulation. Uniformity of weight. In this test, 10 filled containers are collected and weighed individually, and their average weight is calculated. Now, the containers are emptied and weighed individually. The difference between the two weights is the net weight of the contents of the container. The average net weight of the contents of 10 containers should not be less than the labeled amount, and the net weight of the contents of any single container is not less than 91% and not more than 109% of the labeled amount, where the labeled amount is 50 gram or less. It is not less than 95.5% and not more than 104.5% of the labeled amount, where the label is more than 50 gram but not more than 100 gram. If this requirement is not met, then the net weight of the contents of 10 additional containers is taken. Now the average net weight of the contents of the 20 containers should not be less than the labeled amount, and the net weight of the contents, not more than one of the 20 containers, is less than 91% or more than 109% of the labeled amount, where the labeled amount is 50 gram or less, then 95% or more than 104.5% of the labeled amount when it is more than 50 gram but not more than 100 gram. The test is same in Indian Pharmacopoeia, British Pharmacopoeia and United States Pharmacopoeia. Uniformity of mass. The contents of 20 containers are weighed individually, then are emptied as completely as possible, and the average mass is determined. If not more than two of the individual masses deviate by more than 10% from the average mass and none deviate by more than 20%, then uniformity of mass of doses is delivered by the measuring device. The specifications for liquid orals as per BP defines uniformity of mass deviation not more than 10%. pH measurement. The measurement and maintenance of pH is also a very important quality control tool for testing of liquid formulations. Generally, there are two different types of methods used in the measurement of pH. The simplest and the cheapest is to dip a piece of pH paper into the sample, the pH of which is defined as the presence of hydrogen ions in the formulation. The other method is the use of pH meter, which is a device calibrated to measure the pH of the sample through electrode. The pH value of aqueous solution should be taken at a given temperature and only after the equilibrium is reached so that pH drift is minimized. In pH measurement, electrodes are immersed in the solution under examination and the pH is measured at 25 plus minus 2 degree centigrade. The ideal pH of a solution lies in the range of 2 to 7, where most of the drugs exhibit high solubility, leading to their prompt dissolution. Density. Specific gravity or density of a liquid dosage form is an important parameter. A decrease in density often indicates the presence of entrapped air within the dosage form. Density measurements at a given temperature should be made using well-mixed uniform liquid formulation. Density of a liquid formulation is determined using pycnometer or the density bottle. Density bottle is a round bottom flask kind of a bottle with a lid, and it can hold around 25 ml of the liquid for which density is to be determined. In pycnometer, a probe is there which is dipped into the sample holding the liquid solution, and automatically the density in terms of gram per ml is determined for the liquid composition. Viscosity. Viscosity is a property of liquids which is directly related to the resistance to flow. Viscosity measurements are important as for the quality control test in case of syrups and elixirs. Viscosity and consistency directly indicate the stability of the solution. Absolute viscosity, which is defined as the viscosity when all the specifications and parameters are defined, or the relative viscosity, which is determined in comparison to the standard sample. Both viscosities are determined for liquid dosage forms. Sometimes dynamic viscosity is determined, especially for viscous liquids, where resistance to flow is encountered when one layer or plane of fluid attempts to move over another identical layer or plane of fluid at a given speed. For viscosity measurement, Brookfield viscometer is the widely used apparatus which is shown here. This Brookfield viscometer is used to know the viscosity of liquid formulations. It has a spindle which rotates into the vessel holding the liquid solution, and the viscosity of that solution is depicted on the display unit in terms of centipoise or millipoise. Microbial evaluation. Microbial evaluation again is required especially for non-sterile liquid formulations because these are produced under non-aseptic processes and therefore are not expected to be totally free from microbial contamination. Therefore, these liquid compositions are tested for microbiological quality, and these monographs have been established in pharmacopoeia for this kind of evaluation. The major contaminant of non-sterile liquid dosage forms include bacteria, yeast, and molds. There are some oral liquid formulations in which microbiological contamination can present significant health hazards also. Microbial examination is performed by counting total viable aerobic microorganisms using the plate count method. And the media used for microbiological analysis include casein soybean digest medium, agar medium, nutrient broth medium, nutrient agar medium, mannitol salt agar medium, and the blood agar medium. For liquid samples like syrups, oral formulations, nasal drops, usually 10 ml of the product is diluted in a sterile buffered solution with the following composition. For example, containing peptone 1 gram per liter, potassium dihydrogen phosphate 3.7 gram per liter, disodium hydrogen phosphate 7.2 gram per liter, and sodium chloride 4.3 gram per liter. And the medium is maintained at around 45 degrees centigrade in a petri dish. Generally, tenfold dilutions are made for the composition as per British Pharmacopoeia, and in this method 1 ml of the sample is added to 20 ml of the liquefied casein soybean digest agar medium, and at least 2 of the petri dishes for each level of dilution are used. The plates are incubated at 30 to 35 degrees centigrade for 5 days unless a reliable count is obtained in a shorter period of time. Suitable dilutions yielding less than 300 colonies are counted. The arithmetic mean of the counts is taken and the number of colony forming units per gram or milliliter is calculated. While most of the procedures specify a sample volume equivalent to 1 gram, which is around 1 ml of the product, but the salmonella test is the only case where we require volume around 10 ml or 10 gram. For total yeast and mold count, sabuterol dextrose agar medium, which indicates colony formation less than 100 is taken, and this medium is incubated at 20 to 25 degrees centigrade for the yeast and molds, and for less than time period of 5 days is used. The presence of Aspergillus bacillus and the Candida albicans is evaluated in the petri plate holding this medium. As we know that the liquid formulations are evaluated for their microbiological quality, and in this test, usually the dosage form is evaluated for total aerobic microbial count expressed as colony forming unit per gram of the sample, and total yeast and mold count expressed in terms of colony forming units per gram. Most of the liquid formulations are evaluated for DAMC and TYMC. For example, oral non-aqueous formulations should not have more than 10 raised to the power 3 CFU per gram and not more than 10 raised to the power 2 TYMC grams. The formulation should show absence of microorganisms, especially Escherichia coli, that is E. coli. The other kind of oral formulation, including aqueous oral formulation, should not show higher than 10 raised to the power 2 CFU per gram for TAMC and not more than 10 raised to the power 1 CFU per gram for TYMC. This formulation again should not show presence of Escherichia coli or E. coli. The third kind of oral formulation, which includes spray products, also the mucosal products, should not show presence of TAMC not more than 10 raised to the power 2 CFU per gram and not more than 10 raised to the power 1 CFU per gram for TYMC. Again, there should be absence of microorganisms, especially Staphylococcus aureus and Pseudomonas aeruginosa in this kind of a product. Another category of oral liquid dosage form includes dental products, which are also called as gingival products. In these products, not more than 10 raised to the power 2 CFU per gram for TAMC and not more than 10 raised to the power 1 CFU per gram for TYMC should be there. This product also should show absence of microorganisms like Staphylococcus aureus or Pseudomonas aeruginosa. The last kind of oral formulation include the formulation which is to be instilled into the ear, and this formulation is also called as auricular. Here, the number of microorganisms present should not be more than 10 raised to the power 2 CFU per gram for TAMC and not more than 10 raised to the power 1 CFU per gram for TYMC. The formulation, that is eardrop, should show absence of microorganisms like Staphylococcus aureus and Pseudomonas aeruginosa. Stability testing. International Conference on Harmonization, that is ICH Q1A R2 as per Food Drug and Administration, that is FDA guidelines, which are followed for testing of liquid dosage form during a stability program. These guidelines indicate that the analysis for the product attributes that are susceptible to change during storage should be carried out, and the likely influence of these attributes on the product quality, safety, and efficacy should be judged properly. Therefore, parameters such as description of the composition, pH, water content, assay, and total count of aerobic microorganisms, yeast, and molds are analyzed. All these parameters are evaluated at 0, 1st, 2nd, 3rd, and 6th month intervals. The conditions under which these parameters are analyzed include: for accelerated stability study, 40 degrees centigrade plus minus 2 degrees centigrade at 75 plus minus 5% relative humidity, and for long-term stability, 5 degrees centigrade plus minus 3 degrees centigrade and 25 degrees centigrade plus minus 2 degrees centigrade, both temperatures at 60% plus minus 5% relative humidity. These two conditions are assessed for all the liquid dosage forms. The test is carried out in the same container as the final marketed product. After storage for a specific period, solutions with and without preservatives are analyzed for microbial contamination. Water activity. Water activity is the ratio of water vapor pressure in the product to vapor pressure of pure water at the same temperature. Water activity, which is denoted as AW, can be determined directly from the partial vapor pressure or dew point, or indirectly by determination of equilibrium relative humidity. Pharmaceutical drug products with water activities well below 0.75 are excellent candidates for reduced microbial limit testing. When formulating an aqueous oral or topical liquid dosage form, the candidate formulations should be evaluated for this water activity so that the drug product may be self-preserving if possible. For example, small changes in sodium chloride, sucrose, alcohol, propylene glycol, or glycerine in a formulation may result in the creation of a drug product with a lower water activity that can discourage the proliferation of microorganisms in the product. This is particularly valuable with a multiple use product that may be contaminated by the end user. Water activity for some liquid dosage forms. For example, the liquid dosage form which is to be instilled into the nasal tract, also called as nasal inhalants, must have water activity 0.99, and such formulations have the greatest potential contaminant, that is the gram-negative bacteria. Therefore, these nasal inhalants must be tested for TAMC, that is total aerobic microbial count, TYMC, total yeast mold count, and absence or S. aureus and Pseudomonas aeruginosa. Rest of the oral liquid formulations must have water activity 0.9, and these formulations have the greatest potential contaminant, that is the gram-positive bacteria or the fungi. The recommended testing for such formulations include the evaluation for TAMC and TYMC. Labeling. Every pharmaceutical preparation must comply with the labeling requirements established under good manufacturing practice. The label of liquid dosage form is evaluated for the content such as the name of the pharmaceutical product, the name or names of the active ingredients, the amount of active ingredient in a suitable dose volume, the name and concentration of any antimicrobial preservative, and the name of any other excipient, the batch number or lot number assigned by the manufacturer, the expiry date, and when required, the date of the manufacture, any special storage conditions for handling, precautions that may be necessary, the directions for use, warnings and precautions that may be necessary, and finally, the name and address of the manufacturer or the person responsible for placing the product on the market. So students, let us now summarize what we have learned through this module. It is now very clearly indicated that around 90% of the marketed drugs have low solubility. Therefore, to design liquid formulation for such drugs is a mandate. And to design such a formulation, we need to add additives because the drug is poorly soluble. So additives used in liquid compositions include co-solvents or the solubilizers. These formulations offer several benefits, which is why they are mostly liked by the consumers also. However, addition of excipients make these formulations vulnerable to stability over an extended period of storage. Thus, their evaluation involves assessment of qualities such as clarity, pH determination, viscosity, uniformity of content, which is also called as an assay for the formulation, uniformity of weight, density, and the color stability. If we see Indian Pharmacopoeia, British Pharmacopoeia, United States Pharmacopoeia describe most of the methods used for evaluation of these dosage form, but still some of the quality attributes are not indicated in these pharmacopoeia. Thank you.